Good afternoon and welcome to this meeting of the Royal Statistical Society and the PSI Journal Club on the topic of combination therapies. And our meeting this afternoon is sponsored by Quintiles and Wiley. I'm James Carpenter and I'll be chairing the meeting. I'd like to remind you that this afternoon's meeting is being recorded and the recording will be made publicly available on the PSI and RSS websites. During the meeting this afternoon, if you're joining via Global Meet and you experience technical problems, please alert the support team using the chat box at the bottom left of the screen. I'm pleased to say we have two speakers this afternoon. First, Dr. Michael Sweeting will present a paper he published recently in Pharmaceutical Statistics, co-authored with Adrian Mander and entitled Escalation Strategies for Combination Therapy Phase 1 Trials. Michael has worked at the MRC Biostatistics unit in Cambridge since 2002 and completed a PhD at the University of Cambridge in 2008. In September 2013, he joined the Cardiovascular Epidemiology unit, also at the University of Cambridge. His varied research interests include risk prediction using longitudinal and survival data, meta-analysis, cost-effectiveness analysis for public health interventions, as well as novel designs for early-phase clinical trials. Today he's going to present some of his work on dual-agent phase 1 trials, which is focused on the distinct objectives of such trials and the need to often evaluate more than one dose combination in phase 2 trials, leading to more complex dose escalation strategies. Our second speaker this afternoon is Dr. Ying, who will present a paper he recently co-authored in Applied Statistics, together with Chunyan and Yuan, entitled A Bayesian Dose-Finding Design for Oncology Clinical Trials of Combinational Biological Agents. Ying is an Associate Professor in the Department of Biostatistics at the MD Anderson Cancer Center, Houston, USA. His research interests include Bayesian adaptive clinical trial design and missing data. He's the principal investigator of a project for the development of new Bayesian adaptive designs for oncology clinical trials. He's published more than 25 peer-reviewed methodological papers in leading statistical journals on Bayesian adaptive designs, including phase 1, 2, and phase 1/2 trials. The slides for today's talk are available for download from the PSI website, www.psiweb.org, and you can get them by following the link to the Journal Club from the events calendar. Michael's presentation is going to last approximately 25 minutes until 3 o'clock, and then Ying's presentation will follow immediately till about 25 past 3. I'll then invite Tony Sabin from Amgen to open the discussion and the speakers to give an initial response before opening the discussion to all participants. During the presentations, participants' phones will be muted, but these will be unmuted for the open discussion. During the open discussion, in order to keep background noise to a minimum, please keep your phone muted if you are not asking a question. Entering *6 on your phone will toggle muting on and off. We will close the meeting promptly at 4 o'clock. So, without further ado, I'd like to ask Michael Sweeting to give his presentation.
Thank you very much, James, and thank you very much to the organisers for this invite to present at this journal club. So today I'm going to talk about escalation strategies for combination therapy phase 1 trials. In particular, this work is based on the published paper in Pharmaceutical Statistics in 2012, and it's in collaboration with Adrian Mander at the MRC Biostatistics unit. So just to move on to slide 2, this is the outline of my talk. I'm going to first of all start by just talking a bit about phase 1 trials, and then how dual agent or combination therapy trials differ and how the aims and objectives of these trials may differ. I'm then going to present 2 possible parametric models that we might want to consider when modelling a dose-toxicity relationship. But the main focus of this talk is on how we might escalate during a phase 1 trial and what the strategies are in combination trials. So I'm going to talk a bit about admissible doses. These are dose combinations that we may want to give to the next cohort of patients in our trial. And I'm going to talk about decision rules. So these are rules that allow us to choose amongst our set of admissible doses. I'm then going to just present a few results from the simulation study that we presented in our paper that look at these different strategies and round off with some conclusions. So moving on to slide 3, the first aim of a phase 1 trial is really to experiment a new drug or clinical procedure in human subjects. And traditionally the aim is to find a safe yet potentially effective dose for future phase 2 experimentation. And the key here is that the outcome is often a toxicity outcome with some underlying assumption that as the drug increases, so does the toxicity, but also the efficacy of the drug. So we're seeking to find the highest possible dose subject to some toxicity constraints. This is commonly known as the maximum tolerated dose. Now, because of ethical considerations of trialling this new drug first time in man, we usually start at a low starting dose and patients are enrolled in a sequential fashion at different dose levels. So commonly you see adaptive designs used in this field, and Bayesian adaptive designs, for example, the continual reassessment method as proposed by Quigley, are very fashionable and have been used to choose the next dose. So let's move on to slide 4. So the main difference with combination therapies are that, these therapies are becoming increasingly common in the treatment of many diseases, especially in oncology. The designs, although there's many statistical designs out there, the implementation of these designs are still quite naive, and many implementations still fix the dose of one of the agents while dose escalating the other. So reverting back to what we'd call single agent statistical designs by keeping one of the doses fixed. Of course, this doesn't acknowledge that there's any synergistic or antagonistic effects, or does not allow us to investigate these effects. So a more sensible approach is therefore to simultaneously allow dose escalation of both drugs. And really, in combination therapies, the aims and objectives have to differ from that of single-agent trials. For example, there may be multiple maximum tolerated doses. That is, there may be multiple dose combinations that all have very similar toxicity profile, yet the combination of the two doses may be quite different. And so just by looking at a toxicity outcome, we may not be able to distinguish between these dose combinations. And so therefore, you might want to take forward more than one dose combination into phase 2 to assess efficacy. We may also have more prior information. For example, using historical data from the single-agent trials, that's when the drugs are used on their own. And so it would be very sensible to bring in this information in terms of priors into our model. And as I've just alluded to, and as the next speaker will probably talk a bit more about, we may also want to bring in efficacy endpoints as well as toxicity to allow us to choose between dose combinations that have a similar toxicity profile. So moving on to slide 5, well, the first thing we need to decide is a parametric model for our dose-toxicity relationship. And now if you look in the statistical literature, there's many different models proposed. And so the purpose in our paper was just to choose two commonly used approaches and look at how the escalation strategies behave with these two models. And so the two that we used in our paper, firstly, there's one published by Tao et al. In Biometrics in 2003. This is a 6-parameter model, and basically the notation here is the pi x is basically a probability of a dose-limiting toxicity occurring. So our outcome here is binary, whether a dose-limiting toxicity occurs or not. And we want to model this using this potentially 6-parameter model. The reason for using this model is that actually if you set either of the drugs to be 0, that is, you only go back to the single agent design, then this model reduces to basically a logistic model. So, for example, the alpha-1 and beta-1 represent intercept and slope parameters for the log dose for drug A when it's used on its own. And alpha-2 and beta-2 represent the intercept and slope parameters when drug B is used on its own on the log scale. So these have meaningful interpretations. And the alpha-3 and beta-3 can be thought of as interaction parameters that allow synergistic and antagonistic behaviour between the two drug combinations. A more simpler approach, in that there's fewer parameters, is one proposed by Dr. Ying and Dr. Yuan in RSS in 2009. This is a copula-based type model. And again, it has interpretation in that in the margins, this model reduces to basically a power model for the continual reassessment method. So what I mean by that is that you specify a skeleton probability, that's our prior guess of the probability of DLT at the margins. And that's given by these F and G functions. And then the delta function, therefore, the delta parameter gives us the amount of uncertainty around that guess. And the gamma parameter here is used as an interaction parameter. So there are two potential parametric models that could be used. If we jump onto slide 6, you can see that for specified model parameters, we can obtain various dose-toxicity surfaces. And this plot here just shows one potential dose-toxicity surface that was created using that 6-parameter model of tau et al. And in this situation, you can actually see you can get antagonistic behaviour up in the top right, but also synergistic in the bottom left. So you could get many different contours depending on the choice of our parameters. So what do we generally do in a phase 1 trial? Well, we want to escalate from a safe dose and update our model parameters as we learn information. So we would start by specifying an initial dose combination for our first cohort. We would count the number of toxicities that occur. And then, given a certain parametric dose toxicity model with our specified priors, we would update our inferences and obtain a new posterior distribution. And now we want to choose the next dose combination for the next cohort. And this is going to be based on, firstly, a set of admissible dose combinations, and secondly, a decision rule that will allow us to choose between these admissible doses. And this decision rule is generally based on the posterior distribution that we have. And so we continue recruiting patients in this fashion until either we reach a fixed sample size or the precision of a certain quantity that we're interested in is achieved. Moving on to slide 8, I'm going to just talk a bit about what possible admissible dose combinations we could choose. So if you think about in a single agent trial, we often don't want to dose skip. If we have a set of predetermined doses, we want to go up them in a sequential manner, and we don't want to dose skip. Now, in a dual agent setting, that's equivalent to basically saying that we want to move the next cohort within a neighbouring region of where we currently sit. So in this left-hand plot, if we are currently sat at dose level 3 for both drug A and drug B, then we would want the next cohort to be in some region surrounding that dose combination, or to stay at the same dose combination. Now, one strategy is to allow this to happen, but not to escalate both drugs simultaneously. And therefore, we don't allow these diagonal escalations. So this strategy we've called the non-diagonal strategy. On the right, you do allow this kind of strategy. So you allow both combinations to increase simultaneously. So on the right, it's a less conservative approach, and you're allowing faster escalation. A third strategy is to allow diagonal escalation, so allow us to go to any neighbouring dose combination, but also allow us to go to any previously experimented dose combination. So this plot here, the black lines show where we were before, and so we were currently maybe at the bottom right of this grid, but not only are we allowed to go to any neighbouring combination, but also allowed to go back to any previously experimented combination. The advantage of this is that it will allow us to move around the dose toxicity surface more quickly. So, for example, if we've experimented a lot where drug A is given in high doses and drug B is given in low doses, we may want to start experimenting at high levels of drug B and low levels of drug A. And to get to this part of the surface, we need some fast way to get there in the escalation strategy. So given a set of possible admissible doses for our next cohort, we now want to choose amongst them. So we need to specify a decision rule. And the most popular decision rule in phase 1 trials is a patient gain rule. That is, we want to choose the next dose whose probability of toxicity is closest to what our target toxicity level is. So we've got some target toxicity level chosen a priori at the beginning of the trial. This may be, for example, a 30% probability of DLT. And amongst those admissible doses, we want to choose the next dose to be one whose toxicity is closest to the TTL. And that's basically what this formula says here. An alternative approach that you sometimes see in optimal design theory is a variance gain approach. And that's basically saying that we want to choose the next dose combination that gains the most information about the parameters of our dose toxicity model. And a de-optimality approach design does this by maximising the log determinant of the information matrix. And in a Bayesian setting, all we're doing here is maximising the posterior mean log determinant of the information matrix. So this would allow us, therefore, to learn most about the parameters in the model. Of course, if we just use a variance gain approach, we're going to be dosing patients at potentially unsafe doses. So we've got to have a trade-off between patient gain and variance gain. So in our paper, we've come up with kind of like a variance stroke patient gain approach, which I'm going to call here DVAR. And this basically further restricts our admissible dose set. So we need to account for the patient gain in that we want our doses to be close to the target toxicity level. So we're going to have a further restricted admissible dose set, which is the original admissible doses using either a non-diagonal or diagonal strategy. But also we need our doses to have a posterior mean probability of toxicity within a tolerance level epsilon of our target level. So this epsilon is kind of like a tuning parameter which we specify before the trial begins. And we can see that if epsilon is close to naught, then we're returning basically to a patient gain approach. We're basically choosing the next dose that is as close as possible to our target toxicity level. Whereas if epsilon tends to be very large, tends to infinity, then we get to a variance gain approach again, where the toxicity probability for the next cohort is of no interest, and we only want to learn about the model parameters, so we use the Bayesian de-optimal design as it is. So that's the kind of two strategies we're going to be comparing. So we basically conducted a simulation study to look at both the escalation strategies and the decision rules. And what we've done here is we've chosen some prior distributions that were first specified by Tao et al. In their Biometrics paper. Here they were looking at a trial of gemcitabine and cyclophosphamide, and they elicited prior distributions for the probability of toxicity for these two drugs, and their posterior mean probabilities. So their prior mean probabilities are shown in this contour plot, and as you can see, they expected quite a lot of antagonistic behaviour in the toxicity. So we've used those priors for the 6-parameter model, And then we're going to compare how this model performs when we generate data from 4 different scenarios. So the first scenario, we allow the probabilities of toxicity to be in agreement with the prior mean. So we expect the model to perform quite well in this situation. The other 3 scenarios are basically assessing the robustness of our dose toxicity model. So the second scenario, the probabilities are higher than the prior mean. The third scenario, they are asymmetric. And the fourth scenario, the probabilities are relatively flat over the dose ranges. So the actual numbers of these probabilities I won't show today, but they're in the paper. And for today, I'm just going to concentrate on scenarios 1 and 2. So moving on to slide 13. This is the simulation setup. We allowed 6 dose levels per drug. We're targeting a toxicity level of 30%. And for these DVAR designs, we're going to allow a 2.5% tolerance either side of that. That is, we're going to try and select doses that have toxicity either between 27.5% and 32.5%. We've allowed a sample size of 40 and 2 patients to be enrolled per cohort. And as I already said prior, we've set as the scenario 1 probabilities. And we've then run this simulation 1,000 times and looked at each scenario as an admissible dose combination. So just to say here that at the end of the trial, we're going to recommend phase 2 doses based on these following criteria. Firstly, the dose combination must have been experimented on during the trial. And secondly, its posterior mean probability of DLT must be within this epsilon of the target toxicity level. So that's how we suggested to recommend the phase 2 doses. If we have a look and see how the dose escalation performs, and this is the average over the 1,000 simulations, the top graph shows scenario 1. This is where our true probabilities are in agreement with the prior means. And the 3 lines show basically the different admissible dose strategies. The red line is where we do not allow diagonal escalation. And we can see that it escalates slower. It's more conservative. And it eventually reaches the target toxicity level of 30% after about 20 patients. The other 2 designs, that's the diagonal design and the one that allows any previous experimented doses to be reverted to, gets to the target level after about 10 patients on average. So the second scenario, which is the bottom graph of slide 14, the true probabilities of toxicity are higher than we expected in our prior mean, and therefore we do start to see this slight overdosing before the designs revert and converge back to the 30% level. And so you can see that actually with the non-diagonal design, we still get this slow escalation, but it doesn't peak as high, and it takes slightly longer to get back towards the 30% level. If we look at the experimentation, this heat plot basically shows where people were being experimented on in the 6x6 grid over the 1,000 simulations. The first thing to note, if we look at the top row, so the top row is the non-diagonal escalation design. And actually what we see here is that patients are being assigned doses along the margin, and we don't see a step-like fashion up through the doses. And the reason for this is that the slope parameter, our prior slope for the drug A is actually higher than the prior slope for drug B. So the design wants to reach the target toxicity level as quickly as possible, and therefore it tries to escalate up drug A before it starts escalating up drug B. So this is one of the drawbacks of using a non-diagonal approach. You get this unwarranted behaviour. Second thing to notice, if we look at the comparing between column 1 and column 2, these are now the patient gain approach versus the patient and variance gain approach, where we use the optimal designs. And we can see, in general, for the second column, we get much more varied experimentation amongst the doses that are on the toxicity, the target toxicity level. I should say that these 4 doses that are have the black boxes around all have probability of DLT equal to 0.3. So we want to experiment on all these doses if possible. Whereas if we look at the patient gain approach, what tends to happen is that we get stuck in one region and we don't see the varied experimentation around the 4 potential MTDs. So if we look at what the designs recommend at the end of the phase 1 trial, well, we're seeing that approximately about 60% of the recommendations have toxicity within the target range. So that's doing quite well, but there isn't too much difference between the different designs in that respect. What we do see is that the variance gain approaches select more MTDs for selection for phase 2. And the reason for that is because they're experimenting more during the trial, and we can only recommend doses that have been experimented on. We see the similar kind of patterns in scenario 2, slightly better recommendation percentages for the diagonal compared to the non-diagonal approaches. So let me just summarize now. Basically, the escalation strategies for a combination trial are more complex. We have to think about a multitude of directions that we could move from going from one cohort to the next. We've seen in our simulations that if you use a non-diagonal strategy, it rarely behaves in a step-like manner moving up through the dose combinations, and it will generally escalate one margin first and then start escalating the second drug. It may also get stuck in regions where one drug is given at a low dose, as we saw in some of our simulation results. Now, we could use less constrained algorithms. This allows us to be more flexible in our experimentation, but of course this also places more faith on the underlying model. The second thing we looked at was the optimal designs. This allows for more varied experimentation, and that means that more drug combinations can be recommended at the end of the trial. Of course, there's this trade-off between a patient and a variance gain decision, and so this tuning parameter, this epsilon, is one way to allow this trade-off. So if we want to be more specific in terms of the target toxicity level, then we make that epsilon parameter smaller. And of course, this methodology could easily be extended to incorporate other outcomes. So for example, we may be collecting PK/PD information or information on efficacy biomarkers during the trial. And then we may want to select amongst our admissible doses those combinations that perform well in terms of the efficacy biomarkers. So our decision rules could be updated so that we can incorporate these other outcomes. And in particular, we may want to think about decision rules that penalise non-effective doses from being chosen. So that's what I've got to say, but I'll just leave you with those 3 references. And thank you very much.
Thank you very much, Michael, for a very clear presentation. I expect people have a number of questions, but I'd like to ask you to hold those, and we're going to go straight on with the presentation from our second speaker. For those of you who joined during Michael's presentation, Dr. Yuan, who will present the paper in a second, he recently co-authored in Applied Statistics, is Professor at the MD Anderson Cancer Center, Houston, in America, and his research interests include Bayesian adaptive clinical trial design and missing data. He's principal investigator on a project for the development of new Bayesian adaptive designs for oncology trials, and has published more than 25 peer-reviewed methodological papers in leading statistical journals relating to Bayesian adaptive designs. And I think the slides are now loaded, so I'd like to invite Ying to begin his presentation. Thank you.
Yes, thank you. Thank you for inviting me to join this journal club. So today I'm going to talk about, can everyone hear me? So, yeah, today I'm going to talk about Bayesian phase 1/2 design for oncology clinical trial of combining biological agent. So this is part of my dissertation of my PhD student, Chunyan and collaborate with my former colleague, Yuan. So in this talk, I first briefly describe the difference between a biologic agent and a conventional cytotoxic agent. Then propose our dose-toxicity and efficacy model, and followed by dose-finding algorithm. And we will present a simulation study to evaluate the operating capacity of the design and finish the talk with some concluding remark. So page 3. So recently, the paradigm of oncology drug development is expanding from a traditional cytotoxic agent to novel biological agent. So examples including like biospecimen targeting a specific tumor pathway, and gene product aiming for DNA repair, and recently immunotherapy becomes a very hot field in cancer treatment. So page 4. Biological agents are very different from a conventional cytotoxic agent. So for cytotoxic agent, it's often reasonable to assume toxicity and efficacy monotonically increase dose. So therefore, the goal of phase 1 trial is to find the maximum tolerated dose, because that's the dose patient can tolerate and also has the highest efficacy. But for biological agent, typically, the toxicity is usually tolerable within the therapeutic dose range and may plateau at high dose level. In addition, the dose efficacy curve often follow a non-monotone pattern. So therefore, in this case, the goal is to find the optimum biological dose, which is defined as the dose yielding the most desirable treatment effect. So later on, we will see what we mean most desirable treatment effect. And you can see in this case, it doesn't make sense to find the MTD, because the MTD does not necessarily have the highest efficacy. So page 5. And treating patients with a combination of agents is very common in cancer clinical trial. And by combining different agents, and we help to achieve synergistic treatment effect. And many drug combination trial design has been proposed, but those designs focus on cytotoxic agent, and they are not applicable to the trial combining biologic agent because the difference between the biologic agent and the cytotoxic agent. For biologic agent, phase 1/2 trial is imperative. It's because the non-monotonic dose efficacy and toxicity relationship. So for biologic agent, we have to consider toxicity and efficacy at the same time. And in many cases, and the dose finding is mainly driven by efficacy. Next page, 6. So our research is motivated by a lymphoma trial. So in this trial, we combine two novel biologic agents to target the two components in the PI3K pathway. And agent A is a PI3K kinase inhibitor, and agent B is the mTOR inhibitor, which is located downstream in the pathway. And we consider 4 doses of agent A and 4 doses of agent B. And our goal here is to find the biological optimal dose combination, BODC, and which is defined as dose combination with the highest efficacy and tolerable toxicity. So next page. Page 7. Here we show the pathway of the PI3K pathway. And agent A and agent B, you can see they targeting different components in this pathway, PI3K kinase and mTOR kinase. Next page, page 8. So here is an overview of our purpose. So we want to propose a phase 1/2 trial design to identify the BODC, a biological optimal dose combination. And specifically, we use a change line model to reflect the property that dose toxicity surface may plateau at high dose. And we use the logistical model with quadratic term to accommodate the possible non-monotone pattern for the dose efficacy relationship. And in addition, we devised a novel adaptive dose-finding algorithm to encourage sufficient exploration of the two-dimensional space. Because we find this is different from the one-dimensional dose finding, and because we have two agents, it's very easy to stuck in some local optimum. So it's very important to have adaptive dose finding algorithm to encourage you to explore the dose space. Page 9. So here is the basic setup, and we consider J doses of agent A, A1 to AJ, and K doses of agent B, B1 to BK. And we use AJBK, denote the combination of dose AJ and dose BK. And we use PJK, denote the toxicity of the combination AJBK, and QJK, denote the efficacy for the combination AJBK. And as we said, our goal here is to identify the BODC in the J times K dose matrix. Page 10. So here, to model the toxicity, and we use a change line model. So here, if you look at the equation, the PJK is the toxicity of the dose combination J and K. And if you look at the first the equation in the parentheses, so this is essentially the linear term. So it describes how the toxicity increase with the dose AJ and BK. And the difference from standard model is here we have an indicate function. Okay, so you can see if this beta 0 plus beta 1 AJ plus beta 2 BK, if it's less than a certain threshold, so the first term gonna turn on and the second term gonna turn off. And in this case, it's just the standard linear logistic model. And in this case, you can see dose toxic surface monotonically increase with the dose. And once the beta 0 plus beta 1 AJ plus beta BK larger than W, and in this case, the first term going to turn off, and the second term going to turn on. And you can see second term is just a constant W. And you can see after that point, the toxicity essentially keep as a constant. And also you can see when it's reached a plateau, the toxicity probability essentially is given by the exponential W divided by 1 plus the exponential W. And the one thing you may notice here is we didn't include an interaction effect for the two agents. So because we found to estimate such an interaction, we need a relatively large sample size. But for early phase trial, we have very small sample size. And we did the experiment, including the interaction term. We find it doesn't help, and often it make the design performs worse. So that's the reason we ignore the interaction term. And the other comment I want to make is, you may say in some cases for specific trial in the dose range, maybe toxicity monotone increase with dose. Can we still use this model? And actually it's fine because this is a more general model. And because when the W is set to a large value, so essentially you can see toxicity monotonically increase with dose. So in that case, when you estimate W, it's going to be a large value. That means at the investigational dose range, it won't reach the plateau. Page 11. So this is the model for the efficacy. It's a very standard logistic model. And here we put a quadratic term, AJ square and the BK square, because we want to capture the non-monotone pattern. So we allow efficacy increase and then decrease. And here we model the marginal distribution of toxicity and efficacy. You can see we have two separate models. And although joint model is possible, but we have, as we said, so as I said, we have a small sample size, and it's very hard to reliably estimate the correlation parameter. And we did some sensitivity analysis, so it shows model marginal distribution is adequate. Okay, page 12. So here shows the likelihood of the data under our model. Here we use NJK denote the patient treated as the paired dose AJPK, and the XJK1JK patient has experienced toxicity and efficacy. And the marginal likelihood for the toxicity essentially is just to follow a simple binomial likelihood. So similar for the efficacy data, it follow a simple binomial likelihood. And the next page, page 13. So if we assign prior distribution to those unknown parameters, omega, beta, and gamma, and we can easily get a posterior distribution. And in our setting, we use the wake prior and let data speak for themselves. So in here, we use some cozy prior, pretty wake. Page 14. So we have discussed our dose toxicity and efficacy model. And next, we will describe our dose finding algorithm. That is how we conduct the trial. And our design is conducted in two stages. Stage 1 is a run-in phase. And in this stage, we escalate dose along the diagonal to explore the dose combination space quickly. And the goal is to connect some preliminary data so that we can fit our model more reliably. And the stage 2 is more like formal model-based dose finding phase. And in this stage, based on observed efficacy and toxicity data, we continuously update posterior estimate of toxicity and efficacy, and then assign patient to the most appropriate dose. So in order to describe those dose finding algorithm, we first define what's a safe dose. So here we are defining dose AJBK is safe if conditional on the observed data D, its posterior probability less than phi is larger than a certain cutoff. So phi here is the target toxicity upper limit. So that is the toxicity limit we don't want to pass, like 30%. And the delta is the pre-specified safety cutoff. And in practice, we can calibrate this value by simulation study. Page 15. Okay, here it gives more detail about the run-in period. So specifically, the trial starts with the treatment of the first cohort patient at the lowest dose, A1B1. And if the current dose is safe by the criterion we just defined, we're going to ask a dose along the diagonal. And of course, if the lowest dose, A1B1, is toxic, we're going to terminate the trial. And the stage 1 completes when either current dose is toxic or the highest dose combination is reached. And after that point, we're going to switch to the stage 2. So this is just echo Michael's point. So we also found it's very important to allow diagonal escalation to explore the space. Okay, in order to describe our stage 2 model-based dose finding, here we introduce some concepts. And we first define a D-degree neighbor of AJBK as the dose combination whose dose level are different from AJBK, no matter than G level. So essentially, it's just dose close to the current dose. And then we define a G degree admissible dose. So essentially, it's just a safe dose in this G degree neighbors. Page 17. So this diagram shows you can see the black dots is the current dose, and the blue circle are the first degree neighbor of this dose. And the red circle shows the second degree neighbors. So essentially, it's just the dose around the current dose. Okay, so here is how we do the stage 2 dose finding. And essentially, based on the observed data, we first identify the non-empty set of safe neighbor of AJBK with minimum degree of G. So in other words, we're trying to find the safe dose that is close to the current dose. And among the dose in this safety neighbor, we're going to identify the dose with the highest posterior mean of efficacy. So in other words, we're trying to find the dose that has the highest efficacy, which is also close to the current dose. So the reason we want to focus on the dose close to the current is because we don't want next dose transition to jump to a very far place. We want it close to the current dose. Page 19. So here the diagram shows the concept we just discussed. And you can see in the middle, the dot 1, it shows the current dose. And the other dose is the first degree neighbor. Page 20. And you can see those blue ones indicate the admissible dose. And you can see in this case, on the up corners, those two doses are not admissible, so they are too toxic. And next page, page 21. And you can see right now we labeled the estimate of efficacy for each dose. And you can see in this case, the one indicated by red is the dose has the highest efficacy, and which is also admissible. So in most design, and we're going to assign the patient to that dose, right? Because we want to assign dose to the we want to assign the patient to the dose with highest efficacy. However, the problem is go to page 22. So we find that those greedy algorithm is easy to trap to local optimum dose, and for two reasons. Because we have small sample size, sometimes we don't even have enough data to tell us its local optimum, and we already exhausted our sample size. We don't have more chance to explore the space. And the other is due to model misspecification, because we assume some model, but we don't know the truth. And most of the time, we can say in reality, model is always misspecified. And in this case, we may estimate something wrong. And in this case, if we use greedy algorithm, always assign the patient to the current best estimate, we may always stay in one dose without realize there are some bad doses. So to solve that problem, we propose a new dose finding algorithm to adaptively encourage the exploration of untried dose. Page 23. So here diagram shows the main idea. Okay, so in here we still have like 9 doses, and the one with black dot is the current dose. And there are some doses on the side with a person symbol. It shows that dose has been previously used to treat patient. So that means in this lower corner, we treat some patient. Okay. Go to page 24. And you can see, let's assume, based on current observed data, we estimated the efficacy. And the dose indicated by the red is the one with the highest estimate of efficacy. So if we use the greedy algorithm, go to next page, 25. If we use greedy algorithm, we're going to assign the next patient to that dose with the highest estimated efficacy, 65%. But you can see in our case, we don't want to do that because so we're going to assign patient to that one. But in our case, we don't want to do that because in the next dose, the indicated by red, it's the dose have the second highest efficacy, but it has never been used to treat a patient, right? The reason it's lower, it may not, the true efficacy is low. It may very likely because our estimate is wrong. We have biased estimate because of model misspecification. So before we want to continue treating patient at the dose with highest efficacy, 65%, we may want to try the dose, which toxicity is not that low. So because there's maybe chance that the dose is highest, the dose with the highest efficacy. Okay, so we go to page 27. So this adaptive dose finding algorithm can be formally summarized by this slide. So essentially, as we said, the dose J star K star is the dose that has the highest estimate of efficacy based on current observed data. Okay, so the first dot, the first symbol says if nj star k equals to 0, that means the dose, the current dose with the highest estimate of efficacy has never been used to treat a patient, and then we're going to assign next patient to that dose. So this is reasonable, because even that dose have not been used, and we want to try it. But of course, if that dose has been previously used to treat the patient, as we said, we want to explore some new doses. We don't want to always stick in that dose, because there may be model misspecification. And essentially, you can see the estimate, the QJ star K star hat, essentially it's the efficacy estimate for that dose, J star K star. And if we really want to assign the next cohort patient to that dose, it's determined by this threshold. So the threshold is the capital N minus N divided by N to the power of alpha. And here, capital N is the maximum sample size we're going to use in the trial. And the small n is the number of patients that has been treated in the trial. And we can see at the beginning of the trial, the small n going to close to 0, right? So that's why those capital N divided minus small n divided by the capital N is very close to 1. So that means at the very beginning, this is a very stringent condition. So which says most likely we're going to assign the next patient to a new dose instead of the dose has been used to be treating patient. But towards the end of the trial, the small n going to close to the capital N, right? And in that case, this is very close to 0. And in that case, because the late stage of the trial, we already have enough data, and we can trust more about our estimate. And in this case, we have more confidence to assign the patient to the dose we think have the highest estimate of efficacy. So that's the basic idea. And we just repeated these steps until exhaustion of the sample size, and then we select the BODC as a dose combination with the highest estimate of efficacy. Slide 28. So here I'm going to quickly show a simulation study to demonstrate the operating characteristic of the proposed design. So here we consider 4 doses for each agent. So 4 doses for the agent A, 4 doses for the agent B. And the maximum sample size was 15 cohort with size 3. And we set the target toxicity upbound is 30%. And the safety cutoff is 0.4. So this safety cutoff is calibrated by the simulation study. So that's how we get it. And similarly, for the tuning parameter alpha, we set equals 2. Essentially, this alpha controls the threshold we want to do adaptive dose exploration. Page 29. And we compare the proposed design with the greedy design that is otherwise identical to the proposed design, except that it uses the greedy dose finding algorithm. So in other words, it always assigns the next cohort patient to the dose with the highest estimate of efficacy. And this is typically how people do the trial design. They always assign patient to the best one. And we did 2,000 simulated trials under each scenario. Page 30. And here shows the true toxicity and efficacy under the scenario 1. And you can see in this case, toxicity kind of initially increase and then kind of become flat. And for the efficacy, you can see it's a non-monotone, right? Otherwise, the highest dose in the upper corner should have the highest efficacy, but it's not. And in this case, you can see the combination of dose level 2 for the agent A and the dose level 4 for the agent B. It's highlighted by blue. It's our target one. It have a toxicity tolerable because our upbound is 30%, and also have the highest efficacy probability. Page 31, and this shows the selection percentage and the percentage of patients treated at each dose combination. And those subscript essentially shows the percentage of patients treated at each dose combination. And you can see as highlighted by the blue color, and you can see a proposed design have a higher selection percentage than the greedy design, and also assign more patients to the best dose. Page 32, and here shows another scenario. And in this case, the toxicity surface still increase and then flat, and the efficacy surface is still non-monotone. The difference from scenario 1 is that right now the target dose located at different places, as highlighted by the blue dose. So page 33, and here shows the similar results, the selection percentage and the percentage of patients assigned to each dose. And you can see in this case, the proposed design performs much better than the greedy design, has a higher selection percentage and almost double the number of patients assigned to the target dose. Page 34, and this is another scenario. So in this scenario, the toxicity you can see is monotonely increased with the dose, and the efficacy is non-monotone. So this may happen in some cases. And you can see again the target dose is the one on the left corner. It's the blue one. Page 35, and you can see results very similar. The proposed design performed much better than the greedy design and have a higher percentage of patients assigned to that dose. Page 36. So here is a quick summary of our talk. And we propose a design that accounts for the unique feature of biological agent. So specifically, dose efficacy and toxicity relationship may be non-monotone. And the proposed design adaptively encourages dose exploration into two-dimensional dose space. And our design can identify the target dose with higher selection percentage and allocate more patients to the target dose than the greedy design. And as I mentioned before, in the case that the efficacy plateaus, we can also use the similar change line model to model the dose efficacy relationship. Because in some cases, we don't expect the efficacy going to decrease at the higher dose. It may just become flat. And in that case, we just use the same model as we described for the toxicity. Okay, so this paper is published in JRC, recent issue, 2014. Thank you.
Thank you very much, Yuan, for a very clear and interesting presentation. I'd now like to invite Tony Sabin from Amgen to open the discussion. Tony.
Okay, thanks, James. And yeah, thanks, Michael and Ying. I think certainly some very nice thought-provoking work in those papers. Just to start the discussion, really, just some thoughts. So the concept of really exploring different dose combinations to characterize the dose response surface, I think certainly seems a good one. However, despite various methodologies being proposed probably since the late 1990s, and my overall impression is that today I haven't really seen such modelling approaches being routinely used in clinical practice as yet. So to kickstart the discussion, I thought before going into any real specific questions on the individual papers, I'd be interested, firstly to know whether either of you and perhaps even members of the audience have been able to influence the use of such combination model-based designs in practice. And if so, sort of what was the specific question, and what were the operational hurdles they had to overcome? Maybe I could start with Michael.
Yes, we've actually been in discussion with researchers at Cancer Research UK in trying to implement new agent designs. It's still very much in the design stage, so as yet we haven't implemented them in practice. But these kind of strategies for escalation have all come up in terms of discussion and what they would like to see when conducting their trials. So the quick answer is no, we haven't done it in practice yet. I mean, certainly I'm from a statistical department rather than elsewhere in a pharmaceutical industry, but we have had discussions, and we're hopeful that maybe in the next couple of years we will have a trial implemented.
Okay, thanks, Michael. And Ying?
Yes, I think maybe Amgen maybe is one of the place doing most of innovative trial design. We're trying to use it in practice. But I think, unfortunately, in this case, for combination trial, actually we still kind of stay in a methodology stage and trying to develop new designs. I think the main hurdle is because I think in order to conduct such trial and design, so it's very important to have a good infrastructure. So, for example, we have to have a very nice software, have a very intuitive interface, because in here, most case. So sometimes we're trying to convince the physician to use those new designs. The problem is the physician is very busy, and they leave all those data entry, all those kind of things to research nurse. And in order to make research nurse to use it, so your interface must be very easy to use. And if you have to personally, I find it's really hard to teach them to use a complicated software. So even you put it in R function, they still find it's very hard to use. So I think that the main hurdle is it's still we need develop very nice software. And actually, we are in that stage. We have a specific team dedicated to develop such a user-friendly software. So if we go to our department website, we have quite a bit of such a good software. Like CRM, we have very mature and industrial quality software. But I think unfortunately for combination, we have not implemented such a software yet. But I think that we are working towards that direction. Yeah, and also my feeling is a lot of times the trial may be multi-center, and in that case, and even standalone software it's not enough, and we have to have a web-based application. So different sites can enter the data by themselves and get the instruction what's the next dose they're going to assign. So yeah, I work a lot with physicians. So in general, my feeling is most of them are logistical issue. Actually, physicians also quite enthusiastic about using new design, be more efficient. But I feel the most hurdle is the logistical issue, and we need to educate more about the physician as research nurse. And from our side, I think we need to develop more easy-to-use software. So that's my feeling.
Yeah, great. Thanks, Ying. I mean, just to perhaps add a little bit to that. So one of the main issues I hear with implementing these designs, and I think it often drives decisions to use the one fixed dose approach while varying the dose of the other product, really surrounds ethics and feasibility. So, for example, it's quite common to want to study a new biologic in combination with a marketed chemotherapeutic. And you might imagine that chemotherapeutic is dosed at the maximum tolerated dose. Now, when designing such a study, there's a reluctance to use a dose of the chemotherapeutic lower than what would be the known efficacious safe dose, because there's a strong likelihood then that you may be giving patients suboptimal therapy. And this viewpoint really it takes into account the well-known sort of high failure rate among new cancer therapeutics. It also ties in with the fact that at the point in the development program where we're often looking to perform such a study, the information on efficacy of the new drugs is likely to be very limited. And certainly you're unlikely to have any information on what would be your final regulatory endpoint. And also an added point is, from the study feasibility perspective, there then often questions whether the trial may recruit if patients are not definitely being given doses at what would be the minimum standard of care. So I wondered, if it sounds like, Michael, you've had some conversations with your clinical folks. If you've faced this issue in conversation, and what your thoughts are about overcoming this as well.
Yes, well, I suppose in our conversations we were designing a trial with two cytotoxic agents rather than biologics. But I think it's a good point actually that people will be reluctant to go away from the established maximum tolerated dose of a single agent. And I think it all depends really, if you're thinking back to the models that you're putting on these services, about how much prior evidence you have about these maximum tolerated doses at the single agents. And so if, for example, you brought in a very strong prior for one of your agents, then the dose escalation strategy would try and keep quite close to that MTD, at least initially, and you could even start your trial from that MTD of the single agent if you so wished. So I think it very much ties in with how you allow this kind of evidence to be incorporated into your priors for your model. I still feel that that when you're escalating the second agent, with this assumption model, you still get this. You're still going to get an increase in probability of toxicity. So you may go above your TTL. So at some point you'd probably try to reduce the established dose, certainly by a little bit, so that you can increase the second new agent. And I think it's certainly key that you bring in efficacy information at this point as well.
Okay, thanks, Michael. Maybe is there anybody in the audience who's got any experience with this? It'd be very interesting to hear their thoughts, particularly if they've been successful in implementing one of these.
Just as we go to the audience, can I remind you that there are two ways of participating in the discussion. If you're using Global Meet, you can use the Q&A facility to submit a question, which I can see and then read out, or alternatively, ask your question directly. And remember, when you're not speaking, please mute yourself by typing *6 into your phone. Thanks. Would anyone like to open the discussion?
Okay, James. Maybe while people are thinking, perhaps we could just turn to some individual questions on the specific papers as well. So, I mean, perhaps one just starting with Michael's paper. Given that the methods essentially model a dose-toxicity contour, initially, I think it surprised me that the Bayesian D-optimal approach to selecting doses didn't necessarily outperform or was better than the patient benefit method. I was wondering perhaps the rules for the admissible doses actually mute the D-optimal benefit in some way. But, I'd certainly be interested to hear your thoughts on the lack of benefit for the D-optimal approach here.
Yes, certainly the admissible dose rules will constrain your search strategy. So the D-optimal approach, which it normally would want to go to extremes of the dose range, would not be allowed to do that. And of course, you're bringing in patient gain strategies as well here. So you're still trying to keep within a tolerance level of your target toxicity. So I agree that it's certainly within the overall operating characteristic tables, the D-optimal design and the patient gain design look very similar, but then they are both supposed to be targeting that kind of a toxicity level. So in a way, you wouldn't expect there to be too much difference in the overall average operating characteristics. Where you do start to see differences, I think, is when you look at where the experimentation is taking place, and you see a lot more varied experimentation with the D-optimal designs. And this in itself then translates to a greater percentage of recommended phase 2 doses. So that's where you start to see the gains in using a D-optimal approach in this setting.
Does anybody want to raise any questions online?
Yes, I wanted to ask a question. Can you hear me?
Yes. Could you introduce yourself, please? Give your name and affiliation.
Yes, it's Sarah Bray from Amgen. I was just thinking, I think Michael said that you tested 2 patients per dose combination. I was wondering if you increased that to 3 in the interest of gaining more information and progressing the trial along more quickly, how would that affect the total number of patients that were used in the trial? And what you were talking about earlier made me think maybe if you had one cytotoxic agent and one biological agent, if having more patients in each group would enable you to get more information about efficacy as well as safety, and if that would enable you to, after testing the highest doses of the cytotoxic agent, whether you could then move down to a lower concentration of the cytotoxic and explore whether there was still sufficient efficacy with the new biologic.
Yes, I mean, certainly in our simulations we used a fixed sample size, and we stuck with a sample size of 40, but if you wanted to increase the number of people per cohort, say to 3 per cohort, then obviously for that fixed sample size you'd be experimenting at less dose combinations. So you would need to get the same kind of number of dose combinations to experiment on, you would want to increase the sample size, I think, in that situation. I think in general, designs are generally more efficient in that they reach them MTD quicker if you have fewer patients per cohort because you're adapting more often. But of course the trade-off is that logistically they become much harder to run in that you have to reevaluate your outcomes after every patient is in the trial. So I think this is why certainly traditionally you see a trade-off where you get to about 3 patients per cohort. And certainly if you are bringing in efficacy outcomes and you wanted to assess these as well, then you might want to increase your sample size so that you have maybe a 2-stage approach where you get to a certain toxicity level and then you start looking at efficacy outcomes, so almost as a phase 1-2 trial.
Thank you very much.
I think just to continue with that discussion, I mean, one interesting thing that some of these models build in practice is you could view them as making recommendations to clinicians for the next dose combination to use. I think one of the main advantages is it perhaps does give them that option to explore what they want to do in practice, and that perhaps ties in with your comments about not being too prescriptive.
Okay, are there any other questions? I think this is all very, very interesting.
I have a question, if you can hear me.
Yes, I can hear you. Could you introduce yourself and say where you're based?
This is Katie Simonson from Bristol-Myers Squibb in New Jersey. One of the barriers that we face occasionally in implementing both monotherapy and combination trials is the delay in getting either efficacy or safety results from patients. That is, you need to decide where to go for your next dose when the response may be delayed quite some time, or you may have to make a decision based on incomplete data. I wonder if either of the speakers could comment on the effect of that issue on the escalation algorithm.
Yes, well, certainly in the single-agent trials, there's things such as the tight CRM, which allow you to start to make recommendations before you see the outcomes of all the patients in your trial. That's the time to event kind of methodology. Those kind of ideas could easily be brought across to dual-agent trials as well. I wonder actually if Dr. Yuan could talk a bit about, because I know he's very much into the missing data aspects of kind of like delayed responses.
Oh, yes. So I think, recently we have done quite a bit research on that perspective. So essentially, the reason I think we cannot really have a problem when we have delayed outcome is because at the decision-making moment, some outcome is missing. We cannot observe it. So it's very natural to use a missing data methodology to handle it. And I think back to your specific question, and for combination trial, I think last year in the Bayesian analysis, it's a journal, and there is a design specifically handle delayed outcome for combination trial. So if you are interested, I think you can check that paper. I think I don't know if I fully answer your question or not. Yeah, so there already one paper on that.
Okay, thank you very much. Are there any other questions?
Okay, maybe another quick question for Michael. So the choice of starting dose used, I think it was at 1/5 of the maximum tolerated dose of each of the monotherapies. Now, again, kind of on the theme of wanting to avoid giving patients subtherapeutic doses, I'd be interested to hear if you've got any thoughts or suggestions about how to go about choosing the actual dose range. And also, could there be any situations where we could justify starting a bit higher? So, for example, at 50% of the maximum tolerated dose, and then de-escalating or escalating from there.
Yes, I think that's very wise. Certainly within a combination trial, the dose range may be more prescribed in that we have single agent information and we have an idea of where the maximum tolerated dose is for that single agent. So certainly the upper range of the dose range should be quite well established if the two drugs you want to put in combination have been tested previously on their own. We chose just for our simulation this 12% starting range. It was close to what the Tao paper used. I think they chose a 12% starting range. So that's where that came from. But certainly, looking at your prior information on this trial that we simulated from, you probably want to be starting a bit higher, and maybe you'd be wanting to start closer to the margin for one dose, the more established dose, and then slowly escalate the non-established dose. So certainly those kind of considerations have to come into effect when you're choosing starting doses. This is just for an illustrative purpose. So in fact, we didn't, in the simulation studies, we didn't give that too much consideration.
Okay, thanks, Michael.
Okay, we've still got a few more minutes if anyone else would like to ask any questions.
Okay, maybe I'll ask one to Ying and give Michael a rest for a minute. So often the safety risk to patients can be fairly low with some of the new biologics, certainly relative to the old chemotherapeutics. In a situation maybe where you're combining two new biologics, do you think that there'd ever be scope to simply design an optimal design with efficacy in mind, while of course building in certain safeguards to protect the patients? And then perhaps we could also move away from being in a binomial setting as well.
Yeah, so that's a good point. And certainly, we can do that. So I think basically we can just mainly model the efficacy. For example, use the model you like. So if we think it may unimode shape, we may use logistic model with quadratic turn. So I think in many cases, I know for a lot of agents, we may think it's increased and then kind of a flat or plateau. And in that case, we can use the change line model I just described. And for toxicity, I think if we have a strong prior information that the agent is not that toxic, we may just treat it as kind of a second endpoint. We just do toxicity monitoring. So this is more like phase 2 trial. So we focus on efficacy, but we also monitor toxicity. And in that case, I think for practical point of view, maybe we may just use beta binomial distribution and to monitor the posterior toxicity. So we don't want to use the complicated model. We can do that way. And actually recently, we have done some work as exactly you said. So for the single agent biological trial, and I think it will appear in clinical trials. So in that case, exactly as you said, we focus on efficacy. But it's single agent. But for toxicity, we just monitor it. We don't really spend too much effort to model it.
Okay. Thank you very much. Is anyone else? There's still time to contribute to the discussion. I'd really encourage people who are listening to ask a question.
I think the assumption is that the combination agents are given concurrently, but there may be different scheduling. You can give agent A and then delay by a certain amount, then give agent B rather than giving them concurrently. So that's another factor that goes into the dose-escalation paradigm. And I was wondering if there's a model or a class of models that address the scheduling of the doses as well as the doses themselves.
So, Ying, would you like to answer that?
Okay, so I think essentially most of the time I think you can look at a schedule as another dimension, right? So in this case, for example, let's go back, if you have a single agent and you have a schedule, And in this case, it's kind of exactly like the combination trial. So you just treat a schedule. And most of the time, schedule is kind of nested, right? You have less intensive schedule and more intensive schedule. Essentially, you can treat as low dose and medium dose, high dose. So you just treat the schedule as another dimension of dose. And in this case, so if we have single agent, it becomes a combination. And if we already have two agents, it becomes like 2 times 2, 2 times 2. It's like 3-dimension dose finding. So, and a little bit more complicated case is if the schedule does not have order. And in that case, it's a little bit complicated. So because in that case, we cannot put we cannot directly treat it as a dose, because dose, by definition, it has order. And in that case, I think we can treat that dose as categorical instead of a true dosage. And the same modeling strategy can be used. In other words, so for a simple case, if we use a logistical model, in other words, we just need another factor called schedule. And if it has order, we can treat it as exactly as the dosage. And if it has no order, we can treat it as a categorical variable, assign several levels. And then I think most times the same will fall through, I think. So I don't know if Michael has some comment.
Michael?
Yes, just to follow on from that. And actually, one topic of my PhD student was to look at these kind of scheduling of the two agents, where one is given first, and then the second one is given maybe a few days later. And he has been doing some work with Ken Chung at Columbia University, looking at how if the toxicities are observed before the second agent is given, then this gives us some information about the attributable toxicity risks, and that may therefore affect where you escalate. Certainly, if you see a toxicity before you've even given the second drug, then you can attribute all that toxicity to the first drug. So he's doing some work in terms of attributable and semi-attributable toxicities because of this kind of like a delayed or scheduling of the dose combinations.
Right, thank you.
We're trying to write that up at the moment, but it's not yet published.
Okay, thank you very much, Michael. I think we're more or less out of time now, so I'd like to thank both Michael and Yuan once again for their very clear presentations, and everyone who's participated in the discussion, and particularly Tony. And before we all go our separate ways, I'd like to remind you of a couple of forthcoming meetings that may be of interest, maybe more of interest if you're in the UK, I'm not sure. First, the PSI conference is going to take place between the 11th and 14th of May at the Tower Hotel, Tower Bridge in London. Second, there's going to be a joint meeting between PSI, the Biometric Society, and the Royal Statistical Society on Wednesday, the 4th of June at the London School of Hygiene and Tropical Medicine. And this is a meeting entitled Clinical Trials, Past, Present, and Glorious Future, ? Speakers going to include Linda Sharples, David Spiegelhalter, Stephen Sen, Peter Armitage, and Stuart Pocock. And you can register for that online. Lastly, I'd like to draw your attention to the 2014 Royal Statistical Society Conference, which will take place in Sheffield between the 1st and 4th of September. At the end of this meeting, please click on the poll icon to submit your feedback on the meeting. This is very useful to us in planning future events. The next Journal Club meetings, which will be separate for PSI and the Royal Statistical Society, will be advertised by email and on the respective society websites when the details become available. In conclusion, I'd like to thank you all very much for joining the Journal Club this afternoon, and I hope you've enjoyed it and found it worthwhile. I'd particularly like to thank Jack Beebe, Judith Chorlton, Sarah Bray, Anne Cleverly, and Rhiannon Maudsley for their work in facilitating the meeting this afternoon. And on your behalf, I'd also like to thank Wiley and Quintiles for sponsoring the meeting this afternoon. As I say, I hope you found it a useful and enjoyable meeting, and that you'll spread the word about the Journal Club to colleagues and friends. Thank you for joining us, and have a good afternoon.